Welcome to this taped webinar entitled Plants Know What to Do. And you can see that it's going to be a 4-part series. Part 1 is basic biology and processes. Part 2 will be wheat, Part 3 canola, and Part 4 will be peas and microbes. And these will be done using a lot of photographs and diagrams that show processes in plants. And all the photographs you're going to see are basically photographs I've taken. I'll give you a little bit of an introduction as to who we are. My name is Mike Dolinski. I used to be the provincial entomologist with Alberta Agriculture and had many roles after that, including vertebrate pest control, apiculture, uh, things like organic agriculture for a while. And then I retired and joined the private sector. In the last 15 or so years, I've become intrigued by plants and accumulated a library of photographs, both external structures as well as internal structures, roots, shoots, and reproductive systems in plants. And I like to use those to show farmers and agronomists and students or whoever wants to see them about the miracle of plants. Uh, the company I work with, Taurus, is a ag marketing company that operates from Ontario to British Columbia with a spin-off in Australia. And we provide fertility products, macro, micronutrients, as well as biologicals to be used as tools by farmers to help plants be more productive and get a better return for their investment. So that's kind of a little bit of the background. So let's get on to what's going to happen today. Uh, Bit of an introduction done to objectives. I'm going to do a little bit on history here that intrigues me. Then I'm going to talk about how plants respond to the environment because that's sort of important in terms of understanding that. If you want to be better at understanding plants, you better know what the cell is made of, how photosynthesis operates, and how they produce energy, because those three are really major attributes of that lead to really good crop production. Take a look at roots and shoots and transport, nutrients, and stress. When I was a young man in the early 50s, I remember still thrashing at that time. It was coming to an end, and I was just a little kid back then. And it was a lot of hard work, and you could, small acreages. And we skip forward to today, and what do we got? We got seed drills that are automatically loaded with farmers having to lift sacks to put them in the seeder. We can combine now very easily. It's air conditioning, auto steer, and most of our machines. And we produce crops, big crops, wheat, canola, peas, a lot of other crops. And we do this in our climate in Western Canada mostly, and in the northern Great Plains, in about 90 to 120 days through Montana and North Dakota and northern Idaho, where we have fairly stringent winters, to put it mildly. And when we do that, those products that we produce on our land go into livestock feedlots, swine and so on, and to the urban feedlot. And that has big implications for us going forward, because the urban feedlot is where most of the food products go, and the wastes are then piled up. And one of the things that Taurus is involved in is the recovery of phosphorus from urban centers through processing of waste products after the solids have been eliminated. And I think we have to look forward, as we are recycling everything, and how we're going to recycle nutrients, especially hard ones to find in the earth, like phosphorus, zinc, those kinds of things which are used by plants. So we're going to look at cells, photosynthesis, one of my favorites, energy production, and stress management, because I had this mantra that I would use in the last few years. If you look after photosynthesis and you look after stress, the plant knows what to do. And that's how we came upon the idea of the plants know what to do, because that's true. I forgot to include as much as I should of energy production, so that's really key to how we're going to grow a crop as well. You know, the one thing I think about frequently is that uh, if we look at life on this earth, it all depends on plants, because they're what are called autotrophs. In other words, organisms that can produce all their own food using nothing but light, water, carbon dioxide, and what they can get from the soil through their roots and sunshine, photoenergy that drives the whole thing through photosynthesis. And then when we look at animals like us, we either eat plants or we eat other animals to get all the nutrients we need, which are primarily carbon, because we can't get carbon out of CO2 like plants do, and all the nutrients from the soil, like all the minerals, nitrogen, phosphorus, potassium, all the micronutrients. Those are taken up by plants. Otherwise, we'd have to eat the soil, which we don't really want. And that's why we're taking a lot of supplements now, because we think, or many people are deficient in terms of the nutrients they have in their bodies. And that comes from what they're eating. So if you don't like what you look like, better change your diet, because you are what you eat. But that's the interesting story that intrigued me, and I want to share a lot about how plants grow, because it's really important to the future of humanity. But let's go back in time and look at how eukaryotes, or the multi-celled organisms on the earth, have evolved. Started out as bacteria largely. These are sort of anaerobic bacteria and aerobic bacteria, and they met and fell in love, and they had a relationship, and eventually that organism came across another organism that they consumed, and they got the mitochondria out of them. Well, they thought that was a pretty good idea, because we get all this extra energy now using mitochondria. And they went along their merry way here, and eventually there was another sort of marriage that took place on some of those organisms, and they consumed other organisms that had chloroplasts and plastids in them. And this group here, aerobic eukaryotes, went on to become plants with plastids and mitochondria. And the multi-celled organisms on the earth went on as animals, fungi, those kinds of things, and plants, which is the mitochondria. And those are the two major forms of eukaryotes on this earth, which are the multi-celled organisms, and the animals all depend on the plants. Really amazing. I mentioned something called plastids, like the most famous one, chloroplast, right here. That's where photosynthesis takes place. So I want to just point out some of the other plastids that you don't talk about, but they're important, and came along with that. And they have their own genetics. So do the mitochondria. They have their own genetic pool. And over time, some of that has been taken over by the central nucleus in the plant cell, but they still maintain a certain number of genes to function independently, both of them. So these are important for a couple of reasons, and I'll start here. The proplastids are sort of the base, in other words, the stem cell plastid. From that, it produces ethioplasts in plants, which are there before the plant germinates. And they're the precursor to the formation of the chloroplasts. Once the plant cells get above ground, these ethoplasts convert to chloroplasts. Okay, and then as time goes on, some of those can become chromoplasts, which are the bright colors in leaves and tomatoes and things like that. And then eventually the chloroplasts are converted to gerontoplasts, which is the senescence kind of plastid, which leads to cell death and senescence in the plants. In the meantime, there's a group called leucoplasts here, which are root storage cells and seed storage cells. And those have to be built in the plant as the plant is approaching, for example, maturity here. The amyloplasts are where wheat plants store starch, and I'll cover that in the wheat series. The alloplasts are where plants store lipids, and I'll cover that in canola when we talk about canola, because canola is an oilseed crop. And then we'll look at proteins, which are proteinoplasts in all kinds of plants. So they have different mechanisms of storage of their nutrients depending on what they are storing and how they are making them. And here's the story of the plant. The plant takes in CO2, water, oxygen, sunshine, mixes it with nutrients, and then bums off the microbes that recycle the nutrients in the soil. So I've added that to my list of key ingredients for making a plant. Now I got to point out something for the farmers, and I include the horticulturalists as farmers in there and the gardeners as farmers. You want to think about. We can't control CO2 where it's going up, but plants can always use a little bit more. We really can't control the water that comes down in rain. We can do some things with irrigation and drainage, but on the whole, we can't control water. And here on the prairies, where most of us in Taurus work, uh, water is a rare commodity in some areas and an excess commodity in others. So we haven't got much control truly on it unless you got irrigation. We don't control O2 and we don't control sunlight. Hmm. So what do we have to be really good at? We better be really good at nutrients because that's the only thing we control. And it's the nutrients combined with all of these things that influence the microbes. So nutrients are really important. And when I go through individual crops, I'll talk a lot more about nutrients. So in the end, photosynthesis and stress are key. We got to build cells because if we don't build cells, we don't even get a plant. We got to feed every plant, and that's where nutrition goes in, where we're putting fertility products in the ground and as foliars to feed every plant across that landscape. And it's the only way we can sort of level out some of the variability in terms of nutrients. And then we can protect the crop. Those are the kind of the roles of farmers, and these are the roles of plants. And we got to have these nutrients there right from germination through to rapid expansion, and demand changes. And as the plant gets bigger, it produces more roots, more shoots, gets into reproductive stage. And this period going into reproduction is really critical because that's where a lot of mortality of embryos can take place under stressful conditions. But the one thing we got to do as farmers or horticulturists, by the time the plant gets to reproduction, we have to have it pretty well loaded with nutrients. Because at that point in time, uh, uh, the plant changes hormonally. During this period, the basic sink for all the nutrients and photosynthetates is to grow the reproductive parts and grow the leaves and grow the stems and so on. But when we get to reproduction, the plant suddenly changes its behavior, as well as we get a change in photoperiod, which tells the plant it's maturing and time to get on with reproduction. At that point in time, the focus is not on supplying nutrients, photosynthetates to the roots and the shoots and so on. It's now to the seeds. And as a consequence, it starts reducing the amount of energy it is putting into the roots because the roots can't feed themselves. The shoot is what produces the sugars and pumps it down to the roots. And I'll cover that coming up. And then we got to fill it. And we'll talk about that again when we get into more of the individual crops. And this sort of summarizes it here. We get photosynthesis going on here with sunshine. We get CO2 coming in. We get CO2 going out through respiration. We get the plant breaking down the water molecule and kicking out the oxygen as a waste product. And the plant is taking up the nutrients here and mixing it all into that system there. And the roots are out taking in the macronutrients, the micronutrients in a water solution because plants drink, they don't eat. So it's pretty simple. Let's move on there. I mean, we know sort of how it functions, and we and my granddaughter's in grade 4, and she's learned this kind of stuff in school, so it's nothing new. But sometimes, unless it's not in your consciousness, you forget about all those things. So how do plants respond to the environment? Well, generally, we look at this rule. We got genotype or genes times environment times management equal yield minus the variability. The variability means all the stresses on the plants, the variability across the landscape, the variability in the weather and the sunshine and the rain, and you name it, the nutrient distribution in the soil. So let me just go through this. I use this a lot. I think we could all agree that the plant up here is growing in a different environment than the plant down here in the mid slope. And at the toe slope here, we're going to get a different environment. And the reality is a plant here and a plant there and a plant there are all in different environments. Their roots and their shoots, different weather, different temperatures, different moisture, different nutrients, different disease organisms, different everything. And yet every plant is genetically the same. So how do they do that? Well, we'll keep that in the background for a while, but let's take a look at some of those factors, those variabilities. And we always talk about the environmental, eh? The genes are the genes. The plant knows what to do, so we don't have to talk about the genes here in terms of the yield. They'll do their best under all circumstances because that's how they've been programmed. Well, we always complain about the weather. Too much rain, solar radiation, too much or too little evapotranspiration, too much or too little carbon dioxide. What do forest fires do to forest? Relative humidity, wind, oxygen, hail, flooding, you name it. Those are what cause us abiotic stresses. And then we have this part of it of just what are the biotic kind of situations there versus the abiotic ones. And then what are the complicating factors in terms of soil types and pH and topography and all these nutrients here that are scattered through the soil in terms of both horizontally and vertically in the soil profile. Soil aeration, crusting, drainage, slopes, and so on. So we kind of blame weather a lot. Well, let's take out weather out of the equation. So where can we make improvements going forward? Well, we've got regenerative agriculture, for example, is making big waves, and that's involved in rotations and reduction in pesticides and maintaining life in the soil for as long as possible to support microbial activity, those kinds of things. We have diseases there which we handle quite well. We've improved harvest. We are really selective in varieties. Seed quality is pretty good. All of these things are really important and taken into consideration by farmers. But there's a lot of things that are problems we don't do anything about. Like we don't do much about organic matter in terms of improving it and really working at that. Soil pH is a problem. We'll talk about that a little later. We get compaction, which interferes with rooting depth. We get field topography that influences water movement. We get deficiency of nutrients because it's distributed unevenly and we're not putting them back in a way that's really critical. We're making huge progress by mapping soil zones now so that we can variable rate both seeding and nutrient and pesticides and so on. So that's improving in terms of ability to handle that. Soil water holding capacity, we're not effectively measuring water yet. There are some parts of Western Canada doing a good job of that. Soil aeration is a problem because of compaction. Air moves through the soil into air spaces, and as we compact it, there's less airspace, less oxygen in the rooting zone. And that also affects infiltration water. Soil crusting, drainage, and field slopes have to be changed. So there's a lot of things we can do, and we're going to have to do this if we want to improve our productivity. But I just wanted to point that out, that there's a lot of things we can do if we choose to do it to improve things. There are always costs, there's always logistics. We understand that. But as the costs of productivity go up, the land prices go up, I think we're going to have to get better at handling some of these issues. Let's take a look at variability in another way. This is just a trial from Claire's home in southern Alberta, where one of the research scientists just took samples at random hitting across a field, taking a look at the variability within a quarter section, 160 acres. And there's lots of variability in terms of nitrate, all the way from next to nothing to reasonable amounts. Phosphorus, similar things. Potassium, look at the differences in potassium. The average is really good, but man, there are places where it's low and there are places where it's way too much. Sulfate, look at the variability there. And we know that about sulfate. And sulfur now is becoming a deficiency issue in many parts of Western Canada and some parts of Ontario. pH is a big one here in the West. A lot of Alberta soils are acidifying. And when we see a 2-point difference within the field, that's pretty significant. 6 isn't too bad. pH is between 6 and about 7.5 are pretty good under most circumstances. And we'll talk a little bit more about that under pH. Organic matter also varies across the field, which you would expect. But let's take a look across Western Canada. You can see these maps. This is a zinc map. You can see that once we get below 2 inches, because we're basically zero tilling here, that means we're not cultivating and incorporating the surface nutrients into the 2, 3, 4, or 5-inch zones. So as a result, things like zinc, which are somewhat immobile because they're metals, once the roots get down past 2 inches, there's no way we can move that zinc down if we're even applying zinc in there. Manganese is another metal. Once you get below 2 inches, we're below attention levels. This is just a situation in a specific farm here that I work with. 0 to 3, we got 5.6% organic matter, depletes quickly. pH is acid, getting to a critical point, needs limestone in here. So if we just take this sample, it wouldn't tell us that we got an acid problem in the rooting zone. Phosphorus, we get a lot of phosphorus in the upper 2, 3 inches, and then it depletes as we go down. Potassium, the same thing is happening in potassium, a lot up there because that's where we're putting it, and that's where we're getting the remnants of the potassium in the stems and chaff being deposited. Calcium is deficient in many places where the pH is really low. Even though the calcium levels look good, the pH is interfering with availability. Zinc deficiencies are increasing. I did 100 samples of wheat in 2020, of which 54% of those samples showed a deficiency of zinc in the final crop. Manganese levels go down rapidly, and so do boron. And we don't have to look far. Organic matter, phosphorus, and potassium, all as the roots move down into the soil profile, they're being depleted or have been depleted. The pH, as you can see right here around 7, there's not much issues other than with molybdenum, where we get more molybdenum at basic pH than we do at acid pHs. As a whole, if you're in that 6.5 to 7.5 zone, you're in great shape for the macronutrients. As you go in either direction, some of them become less available. Most of the micronutrients are more available as the pH goes down and less available as the pH goes up. And some of these nutrients, especially phosphorus, as the pH actually drops and it becomes acidified, it then solubilizes things like iron and manganese and aluminum and ties up that phosphorus so it becomes unavailable to the plants. And as we get over pH is about 8, 8.5, it really starts interfering with uptake. And we know that that affects the economics of fertility and the availability of nutrients. Once we get down below 5, we're starting to really affect the productivity of those soils and the efficiency of fertilizers that we put down. Nowadays, there's a lot of talk about this whole system, soil, plants, and microbes, and it's all driven by water. Without water, the plants, the microbes, everything kind of suffers. There's no way about it. But it's now becoming fairly commonly accepted that the plant exudates influence the microbes, and the microbes influence the soil in terms of recycling of nutrients. So this whole combination of plants affecting soil and back and forth, this integration of these is really, really true. And now that we can measure soil microorganisms using metagenomics, trace genomics now provides that service through Taurus here in Canada. And now we can get quantitative measures of both the good guys and the bad guys in the soil profile and predict certain things about risks from diseases and also risk from impacts on recycling of things like phosphorus and nitrogen and other nutrients. So we're going to take a little bit of a look at this. And if we look at exudates, you can find these kind of tables. We get amino acids, organic acids, sugars, vitamins, all kinds of enzymes, and inorganic ions and gases that are in those root exudates and some other kinds of purines and nucleotides. So the plant is pumping in nutrients, these organic compounds into the soil profile in response to stresses and deficiencies to try and extract nutrients from there, provide protective mechanisms in terms of some of the organic amino acids that are used, like proline and some of the hormones that are used to protect plants. So these plants are pumping this into the soil. For example, amylase, which breaks down sugars, can be used to break down sugars in the soil profile and also feed the microbes, because it's these compounds that the microbes need as well. They're after nutrients. They're after carbon. They're getting carbon from the organic matter and from these exudates. So the root exudate is fundamental in terms of plant nutrition, and I believe they control the organisms that they want there by the exudates that they provide. We know that canola is behaves like a weed. It can extract nutrients and produces acids, for example, that will extract products from the soil like nothing else does. And here we know also now about microbes that they elicit systemic resistance in plants. They have an effect on the physiology of the plant, enhance growth. They are involved in enzymatic activities below ground and enhance ability to deal with abiotic stresses. And they produce some phytohormones as well that are useful to the plant and influence that. They suppress soil-borne microorganisms, enhance fertility uptake and nutrient uptake through the recycling of these minerals from both the organic material and from the soil particles in the soil to solubilize those. We even have work that shows that plants, what is termed rhizophagy, will take in bacteria in the root tip and extract the nutrients from them and then kick the shells out so that the microbes can in fact reorganize and rebuild themselves, so they can extract them directly from microbes. If you want to read more, just look up rhizophagy and you'll find a couple of papers on it. Phosphorus is always a key problem because it's so tied up in the soil by calcium, aluminum, iron, and organic products. So we know that there are phosphorus solubilizing microbes, that's what that stands for, that will produce organic acids and polysaccharides and hydrogen that will dissolve these and pull them off with the cation exchange here, the cations. And then the plants put out phytases, which break down the phosphorus or mineralize the phosphorus from organic matter and release them into the so-called pea pool, which is then available to the plant. And we know that they influence nitrogen cycling here. And a lot of you have looked at this and shaken your head about, understanding this. And it took me quite a while to get it. And I just show this to show you that the microbes are important. Nitrosoma, nitrobacter that are involved in nitrification, the conversion of ammonium to nitrite to nitrate, and then to denitrification and loss if it isn't handled properly. And we can see here, just to emphasize that, here are all the microbes that are involved in all the different cycling processes here in the plant, and then we have fixation and mycorrhizae and so on. So in order to help the plant in this situation, when we're putting down urea or UAN, whichever, which is converted to ammonia, and then it's stable there in a positive form here in ammonium, then the ammonia will volatilize and is a problem. And then the ammonium is converted to nitrate. Well, we can stop and slow this down, and we can slow this down so that the plant can take up all the nitrate if it's released slowly, so that there's none left over to denitrify and reduce our nitrous oxide emissions. So we can provide those stabilizers in here that interfere with the microbial activity, and we can measure those quantitatively and determine which fields are at risk. And these are just more. The amount of data coming out now on solubilizing microbes is staggering. Never mind just the phosphorus solubilizing. We got potassium solubilizing, selenium solubilizing, zinc solubilizing. We now can measure and identify the functions of these organisms, and we will eventually, like we are with some of the phosphorus solubilizing ones, uh, be able to supplement the soil and reseed the soil if it's short of those microbes. And just to show you, here's a readout in terms of a trace genetic analysis for total mycorrhizae, which are fungi that evolved with plants. So you can see across all these fields in Western Canada, some fields have massive amounts of mycorrhizae, whereas other fields don't. So we'll be able to make much better decisions about where we supplement mycorrhizal populations and where we don't. So we'll just leave that there. There's nothing much more I can say about microbes until we get to the end when we look at specific products, but they're becoming more and more important in terms of understanding what's going on in the plant. So here we are back to how does the plant interact with the environment besides the microbes? And this is at the plant level, and it's involved everything. First of all, the plant detects a signal. We'll hear salinity, cold, whatever, a signal. Perception of the signal is transmitted, so it moves it down through membranes and proteins, and eventually we get sensors that lead to transcriptional control. In other words, genes are excited, or they're expressed. That's the term I was looking. I always call them up and down regulated, but genes are expressed, which then produce, uh, they're transcribed, and they then produce gene activation, which produces osmoregulators. We produce chaperones. We would produce aquaporins and ion transporters. We produce things like these, and I'll get to them, sods and so on, to detoxify things. And then they go on to provide tolerance and adaptation. Homeostasis is the secret words. The cells are always trying to get to a balance. When they're in balance, they're healthy, they're expanding, you get a crop. When they're not in homeostasis, that means they're under stress. There's something wrong. No different than us, no different than us. And the plants can develop as best they can mechanisms and proteins and enzymes that will deal with those, and we get adaptation. So we do that with genes. Well, here's DNA. Very simple. The P stands for phosphorus. These little boxes are sugars. Deoxyribonucleic acid. And these are nitrogen compounds across here that link the two helixes. And hydrogen is involved. It was sort of the basics that nature had here to work with. So let's take a look at wheat, canola, brassica napus, and peas compared to other plants and the human genome. Human genome here is about 24, 25,000 genes. There's sort of things that go on uh, beyond just the initial translation and protein production, but we won't worry about that. But look at wheat, 107, almost 108. Wheat has a huge, huge genome of the entire wheat genome. Brassica, also tremendous number of genes. Peas, not too bad. Uh, you can see Brassica rapa, the old Polish types, weren't very good, and we got rid of them because they had a small gene pool, so we couldn't really utilize uh, the gene pool that was available to Brassica napus. And we've made huge progress in napus. The rest of these have, varying levels of genes, but if you can see that wheat and crucifers are much more genetically variable than the human being. And they did that through evolution because they were here from sort of developing from plants for a long, long time. And in the plant itself, with all these arrows that it's monitoring what is going on in all these different components of, transport of nutrients, biosynthesis and growth and reproduction and so on, and how the energy production is going between photosynthesis and the mitochondria producing ATP. And the genes are responding during the entire developmental stage and, and are measuring the environment at the same time, and they're doing it with genes, genetically, up and down regulating those genes to make what they need. And they have those transporters that the genes are involved in terms of moving things, and we get secretions and we get biosynthesis that takes place. And those two are basically, biosynthesis just means it can just make, based on the genetic sequences, the enzymes and chemicals and proteins that they need from amino acids and so on. So the plant knows what to do. Now, to complete this, let's take a look at the cell, photosynthesis, and respiration. And it's all about the cells. He who builds the most cells wins the race. The plant that can build the biggest, healthiest cells will be the best producer, the biggest producer. There are two kinds of cells generally in plants. There's called the tip growth cells, and there are two of those. One is the root hair, which is a single cell, and the other one is the single cell is the pollen tube. And I'll talk about the pollen tube when we get into crops in terms of reproduction. The other one are diffuse growth. In other words, they're they're expanded in specific directions and usually vertically or longitudinally because the plants grow upwards, roots go downwards, so you want those cells to be growing in specific directions, and they handle that again genetically. But here are some cells. This is uh, the tip of an emerging wheat plant. And you can see here that the cells are small and over here they're bigger. So the plant moves through uh, both the soil and the roots and at the shoot by expanding these cells once they're developed. You can see here are some stem cells right here. So they're growing longitudinally. And here were some I found in the background of stripe rust. You can see them here in the leaf as well. They're growing longitudinally here to move the leaf out. This radicle will eventually open up to allow the cotyledons to leave, but this is what protects that coming through the ground. Again, wheat will cover that. Within the cell, as I said, we want to get to a state of homeostasis. So you can take a look here, and all those nutrients that are available to the plant are in each cell. Here is chloroplast, iron, copper, magnesium, and you'll see why those are important later on when I talk about chloroplast. In mitochondria, we get iron, phosphorus, potassium, other things there, the nucleus, the peroxisomes, we got some manganese and iron involved. You can see that all these metals are called transition metals because they can change their valence and they're used in oxidation reduction processes. If we take a closer look at a cell though, we'll look at this side first. Here's your chloroplast, and you can see these little circles here like dollars are called thylakoids, which we'll look at in a little while. So on the nucleus, there are sensors here that are picking up sensing in the nucleus, and then they can respond here and move materials throughout the plant to coordinate an action. There's a vacuole here which stores nutrients. Here are mitochondria attached to this little river here that goes around everywhere and things are attached to, so the plant can move things around. Here are protein bodies. Here are oil bodies. So the plant produces oils like a canola plant or an oilseed plant as a secondary metabolite and then fills the seed. If we look closer here, we have the blue lines called the plasma membrane in here. And I think that's about all I need to show you. Uh, that's important. I will make one comment here about the vacuole. The vacuole is like a fat cell in a human being. You have so many, and when you get fatter, they get bigger, and when you shrink, lose weight, they get smaller. The vacuole is the same thing in each cell. Uh, the vacuole can expand to fill about 70–80% of the cell because all of these, what are called organelles, are sort of floating around in a solution called the cytoplasm. They're floating around there, so they can be moved around. So the vacuole, when a plant's, for example, root hits a side band or a mid-row band, there's an excess of nutrients there, and it can almost be toxic in the plant. So the plant, knowing that the future indirectly will have some stress in it, will overload itself and store it in the vacuole. And then as time goes on, it'll release the nutrients out of the vacuole back into the cytoplasm here to feed the rest of the organelles in that particular cell. And that is kind of an amazing feat. Now, to do that, it has to control everything going out. And that plasma membrane that I showed you in blue, these are phosphorus ions here, and a lipid bilayer in here. It's called the phospholipid bilayer. And humans have the same plasma membrane. And within that, there are these transporters and sensors, little antennae that are sensing all kinds of things in there, what's going on. And the plant has to control what's going in and out of the cell. So you can see here that some things can pass through easily, like CO2, N2, oxygen are permeable, alcohols are permeable. Urea and water are somewhat permeable. Urea's got no charge, so it will go through the cell walls and the plasma membrane very easily. Sugar, it's impermeable. The cations and anions are impermeable. Amino acids and nucleic acids are impermeable. Plant just can't let things flow through just here, there, and everywhere whenever there's a high concentration or not. It's controlled. So this summarizes it. Here's the plasma membrane, and you can see all these transporters in here. This is for amino acids. This is for sucrose. This is for sodium, and so on. And then here is the vacuole, which also has a membrane called the tonoplast with all those proteins in there, and a pH that's low. It's acidic in there. So it stores the nutrients that are excessive, and then as it runs out here, these proteins will pump them back in. And eventually, when the vacuole runs out of nutrients, you'll see deficiency in the plant. By then, it's kind of tough to recover, to tell you honest truth. And just to show you, these transporters are now stuck in the roots, so we can pull up nitrate, ammonium, and sulfate. Right here, I can read this a little better. Nitrate, ammonium, and sulfate, plus all kinds of other transporters. They have something here called high affinity transporters and low affinity transporters. All this means, and you can see it uses ATP in here to some degree, to move nutrients across this plasma membrane from the soil into the plant. When there's a lot of nutrient available, it can use low affinity transporters because it doesn't have to do a lot of work, doesn't have to use a lot of ATP. When the nutrients are short, the plant will use high affinity transporters. In other words, there'll be stronger acids, stronger exudates to try and extract those nutrients. And here's where the plant is putting out those carboxylates, those phosphatases to break down phosphorus off of root particles and out of organic matter, and the acids that are going to kick the phosphorus off the cation exchanger, the soil particle. And you can see it's pumping hydrogen into there. And that's why just growing plants will acidify the soil. And that occurs if you haven't got enough limestone there to neutralize that. That's just the way it is. And then we can get things like aluminum and so on kicked off. And these transporters control what's going in and out. And water is controlled through transporters called aquaporins that are there in the cell walls and the roots and throughout the plant. Next, we're going to look at photosynthesis. We know what the cell is. We have the components in it. The plant knows what to do. It's got the nucleus in there. It can make all the ingredients it needs wherever it wants to, and it communicates hormonally across the plant. Photosynthesis, simple. Carbon dioxide plus water plus light gives you sugar and water. That's all photosynthesis is, right here. Carbon dioxide, water, light, glucose, oxygen, and the waste product is oxygen. That's kind of cool. Lucky for us. So this is looking at it. Those little discs I showed you called thylakoids are right here, and inside of the thylakoids right here is where the action takes place. But first thing that happens, the water molecule has to be broken down and the O2 is kicked out as a waste product. Some is retained. And then from there, it produces some ATP, adenosine triphosphate, or energy. And this is also transporting some of this hydrogen over there into the Calvin cycle where CO2 comes in. It runs through a bunch of processes and it kicks out sugar. That's what photosynthesis does. All this ATP produced there is kept within the thylakoid and is not, in fact, used for anything else but other this photosynthetic process. And here's chlorophyll, magnesium, nitrogen. Most of North America is deficient in magnesium. Probably 75% of people are deficient in magnesium because we're not eating enough green leafy vegetables, because there's magnesium in all the green leafy vegetables. We're eating processed foods which don't largely contain all the magnesium, and it's also got a lot of nitrogen, which is used to build proteins in your body. So when the plant is going to senesce, it kills all the chloroplasts here and extracts these nutrients and remobilizes them when it needs them. You don't have to memorize this, by the way, folks. You can go back and you can't see, you will never see any of this inside the plant. So you just need to sort of understand what's going on here. So the sun shines on the plant and it absorbs red and blue lights here, and it excites these little transporters here called electron transport. But here's the key thing. Here's water broken down, and the plant wants the hydrogen. The oxygen is the waste product. Remember what I said that? Thank heavens for us. We get a little electron transport moving along here, and it's kicking in more hydrogen. And that's all concentrated within the thylakoid here to drive ATP synthase. In other words, this is like a windmill. This going through turns this protein and hooks a phosphorus ion to ADP to produce ATP, which then goes over to the carbon cycle and takes in CO2, turns it out, and produces sugar. That's it. Just sugar. In other words, the world runs on sugar because that's the major source of carbon that we get directly from sugar. And then we get the carbon from the animals we eat or the plants we eat as they create sugar from the CO2. The plants do produce sugar from CO2. Now, here's where it's important for farmers to understand this, because here's process H2O is broken down and it wants this hydrogen again to drive this ATP synthase. But along this electron transport, there are some key things, this thing called plastocyanin and this thing called ferredoxin. Ferro means iron. Plastocyanin is basically copper. And I'll show you that kind of stuff. Here is what breaks down the water molecule. The first thing that has to happen is the water molecule broken down to get the hydrogens, done by manganese calcium oxide, that cluster right there. Plastocyanin, copper, nitrogen, sulfur. About 30% of the copper in the plant is used specifically in plastocyanin in the electron transport. Iron and sulfur are always hooked together. The iron is used in ferredoxin in that second phase of electron transport, along with a lot of sulfur, cysteine, amino acids there. I point this out just so you sort of get a grip of why the micronutrients are as important as the macronutrients. If you don't have those, you basically will not have a really well functioning plant. It's just about that simple, to tell you the honest truth. The second thing we're going to talk about is mitochondria and the role of mitochondria in the plant, the powerhouse of the cell. I already talked about energy production of phospholipids, nucleotides, and amino acids, and metabolism. Stress responses, anything that takes power, that has to deal with things. Development, everything that's going on in development in terms of plant cell death is even orchestrated by reduction in mitochondrial activity. In fact, within human health, uh, there's a sort of a concept in terms of mitochondrial health and human health, and poor mitochondria are being linked to chronic disease in human beings. And the plant sort of goes around and kills cells that are weak and low, have poor functioning mitochondria. So that's the way that they can scavenge some extra nutrients by killing off old cells, just like plants do. And it's involved in intercellular signaling, you know, because if the mitochondria are suffering and the sugar is suffering, a signal is sent. Whenever there's a distraction or an issue, there's a signal that is developed, and most of the signals are transmitted by bursts of calcium. Calcium is really regulated in the plant cell, and when the plant has a signal, it'll push out a burst of calcium, and that will trigger a chain reaction leading to largely a genetic upregulation of genes. And here we go again. Where are we heading? They're involved in producing cellular homeostasis because the energy is required to do that. And they change shape and they multiply by binary fission. In other words, it just splits in half. It has a division system like a bacterial, multiply very, very quickly and split. Same with the chloroplast. They multiply by binary fission because they probably came from bacteria. So they have the same process of multiplying. Now, one thing I'll point out kind of right now is that the mitochondria have to be transmitted from one generation to the next along with the plastids because the plant can't make them without the original source being there in the seed. So we'll show you that in a minute. And this is just electron transport in the mitochondria. The same thing, we're developing the hydrogen concentration within the membranes to drive ATP. It's no difference. It's just that the mitochondria use the sugar produced by photosynthesis right here. So that sugar is transported to the mitochondria and goes through the electron transport chain there, feeds that and produces that ATP that we all want to see. So it's these two things that drive the whole plant. The whole plant is dependent on that. If one of those two fail, or both fail, or are both stressed, the plant is in real trouble. And here's some real mitochondria. And this is just for the chemist in the group. This is ATP, adenosine triphosphate. And you can see the 3-phosphorus here. This one splits off, and that leaves energy behind. And then they have to reattach that P to gain the energy back. And when it leaves it behind, that's called phosphorylation. And this summary, I guess I could have just had this one slide. Phosphorus, or photosynthesis, produces sugar. The sugar goes on to respiration. Sugars go on to produce all these amino acids, carbohydrates, and transpiration runs the whole show, producing, and we get the lipids as well, the lignans, everything. And then we translocate those nutrients around the plant. That's the cycle that drives everything with these inputs and these guys as partners. And if anything stops, the whole process goes into a crash mode. Just want to point out one thing about seeds here, and that's I maybe mentioned it a little too early. When a seed is desiccated going into maturity, the mitochondria is damaged. So you can see that as soon as water comes in, the first thing that happens is the plant produces from, again, in wheat, we'll cover this in real thoroughly, but it produces the precursors to repair the mitochondria because it's damaged as it goes into drying, so it's got to be repaired. So the first 20, 30, 40 hours are spent repairing mitochondria and then building and synthesizing mitochondria, because without those, the seed hasn't got the energy to, in fact, go on to germinate. And then as it moves through, because these have to multiply and build from this small base in the seed to grow to massive numbers as the plant goes through that rapid early growth phase, the rapid growth phase, and then to maturity, all starts with the seed. So the seed is really important to the end of the crop. Let's look at transport. Simple. Plant picks up water and nutrients, moves it up the phloem, or xylem, sorry, into the leaf, all the way up to the other leaves and up to the apical meristem, runs it through photosynthesis, kicks out oxygen, moves nutrients, photosynthetes into the plant, and away we go. And it cycles through, and it goes up to the leaf. The leaves right here, I'll show you this. This is a cross-section leaf. Here's the xylem and phloem bringing up nutrients here. These are spongy cells here, and water goes into a vapor phase, and water leaves through the stomata. At the same time, carbon dioxide goes in, and oxygen goes out. And it's the evaporation, the loss of water, that puts suction on the root system and develops root pressure that keeps it going. It's like a siphon that keeps going. And when it reaches the end of the leaf tip, some of it will sometimes be kicked out at the leaf tip, but basically that's the whole process. And these stomata are rigorously controlled, and the guard cells have their own chloroplasts because they're so important. And these are water molecules leaving. The pickup of the nutrients in water occurs largely through the root hairs. It is the root hairs that have a life of 3, 4, 5, 6 days that are responsible for most of the pickup of the nutrients as the root and the lateral roots are going through the ground. So the nutrient goes in directly into the single-celled root hair. Remember, I told you they're single-celled, and here's your vacuole. So it goes in. This is called the symplastic root, and it goes through those plasmodesmata I talked about. That's why I brought it up in the cells. It goes from one cell to the next cell to the next cell to the next cell to the next cell, and eventually it gets to the endodermis right here, where there's this impenetrable strip called the Casparian strip. And then it goes into there, and it can get by the strip and into the xylem. Or it can go between the cells in here and here and here, but eventually it has to go into the cell before it can get into the xylem to move up in the plant. And the plant can store nutrients in here, but it can't make sugar. So the shoot has to ship the sugar down because the root can't feed itself. Now here's this Casparian strip in rice. It's got the best one because rice has 2 Casparian strips because the roots live in water, so it has to keep things out. And it's impermeable. Here it is in barley. And you can see here's this root, the xylem and phloem, the vascular system, and here's the outside dermis of the plant, rice plant. Now at the other end of it, the apical meristem at the top of the plant, it's signaling the root, because the two have to grow together. As the shoot grows, the root has to expand to develop the capacity to feed it. Now here's in canola, these are the cotyledons at the base of the cotyledon. This is the first true leaf. This is the second true leaf. So this apical meristem is where the sort of germ cells are and produce all the reproductive, vegetative parts, and then eventually the reproductive parts. And this tip is communicating with the root apical meristem through hormones, things like gibberellic acid, abscisic acid, auxins, you name it. Here's a pea. This is the apical meristem in pea. Inside that clam leaf, you will get a lot of activity going on here where the germ cells are. And you can see that within that clam leaf, there are already a whole bunch of other leaf components and reproductive components that are going to be developed. So quite frequently, we don't put on micronutrient foliar applications in time because we need to support this throughout the growing period. And if the plant can get the nutrients through the root system, we have no choice but to try and get some on either foliarly or broadcasting if it's able to get into the soil. And this is the tip of a wheat plant. So the plant extracts nutrients by putting acids into the soil, including hydrogen, and kicks off the cations off the cation exchange. And then those are picked up by the root in the flow of moisture, or if the root runs into potassium, for example, or phosphorus doesn't move a whole lot in the soil. Certain nutrients move with the water, the anions, the negatively charged ones, the nitrate, the borate, the molybdate, the sulfates are all water soluble, so they will move with water, and as the root takes it in, there's a deficiency by diffusion, the water will move to it and bring the nutrients. But things like potassium, phosphorus, all the transitional metals, you either got to bump into it or there's slow diffusion in. But that's basically the mechanism that the root is using, and those are the exudates we talked about earlier. Once it picks it up, goes into the xylem, up she goes into the leaf, water out, taking the nutrients through the whole system. And the water is controlled through the stomata, the opening and closing of those little holes, and that's largely regulated by potassium and chloride. And it's water moving in and out that causes that to open and close. And that's why water is so darn important in the plant. It manages the release of water. It manages the movement of nutrients into the plant, but it also manages the movement of photosynthates or sugars in the plant. And they have their own root system that grows at a specific angle. You can see that all these wheat plants, it's not just random. The roots are all programmed to grow at a specific angle. And with time, they can change that angle. Here's a typical root. This is a wheat root that I shot. Here's the meristematic tissue here, just behind the root, and there's a root cap that protects the root sometimes when it's going through the ground. This is the area of elongation. In other words, the cells are being expanded by hydraulic pressure using primarily water and potassium, which moves and drives the root through the ground. It's driven by hydraulic pressure. The root hairs develop after the elongation zone, otherwise they'd be ripped off. And all these exudates here then feed the microbes that are going to be growing back here as the root goes through the ground. And eventually we get lateral roots growing there as the plant is expanding, so the plant can now explore new territories. Here's the tip without the root cap on it. And the root cap is really important in terms of what it does behind. This is a photograph of what I just described. You know, it's a signaling agent and controls mitosis, which is what expands the cells at the root tip, controls gene expression, stimulates microbial growth by the exudates, releases chemoattractants likely to attract those microbes in the soil that it wants. It synthesizes defense molecules. It does a lot of stuff in the plant. It's really key to uh, a lot of the material that work that goes on in the rhizosphere. And this is what's called the rhizosphere. You can see this is a wheat plant uh, root here. You can see the xylem and phloem, and that's where the plant can extract nutrients in contact or by interception or by diffusion. Uh, right in that small zone. It's a very small zone. The actual roots don't contact a lot of soil if you look at it. And you can see this, the root cap, and here's the mucilage that is exuded at the root tip, and you can basically see the vascular system of the plant. This is faba bean. You can see that the lateral roots are developed right here based on the vascular system, slowly push out, and you can see here the vascular system, and expand, come out. And then if you look at them inside, you can see that the lateral root vascular system then contacts directly into the xylem and phloem. So once the nutrients get into the lateral root, they're free to flow. There's no Casparian strip or anything to stop them until they get in here. So it's got a really great mechanism. Here's a canola plant, 7 days old, extracting nutrients from crystal green, which is insoluble. And you can see that the plant has popped out a whole pile of lateral roots. It knew where it was, and it starts melting this crystal green granule by exuding citric acid, malic acids, which will dissolve that inorganic material. Here's roots showing where the sideband is. They sense likely, in my guess, is the nitrate, which is negatively charged and mobile in water, picking up nitrogen because the plant needs nitrogen and phosphorus at all costs. So the plants know where to go. The plants know, as the title of the seminar started, the plants know what to do. From a farmer perspective, I want to point this out as something to think about because you hear about salt index and root toxicity and so on. And this is just an example of the kind of stuff I do. Just showing here how this granule, which has a salt index, and we won't go into what that really means, but it can cause some problems in terms of germination by extracting water from the seed, for example, instead of allowing water to get it, but it also can be toxic. So you can see that a granule at the opposite end from germination has a relatively minor effect on the seminal roots, whereas you put a granule at the germination end and it really impacts the root development because of that salt index likely. And it's just more or less if you're putting salty, and pretty well all our fertilizers are salty to some degree, but if you're putting high salt fertilizer, and potassium chloride is one that's probably the highest salt index of all, it actually can be somewhat dangerous, and it's just a matter of sheer luck in some respects as to where the granule lands, as to how much of an impact it will be. And moisture sort of nullifies a lot of that salt effect, but you can't be guaranteed of water all the time. This is boron and what it does to roots. This is canola root. In presence of a boron granule, it's very toxic to plants at high concentrations. It's only of concern if you're putting in 10 or 15% granules right in the seed row. Put it in the side band or you put it in the mid-row band, it's a lot less. But because it's an anion, that means it's negatively charged and will move with water. You can just broadcast it even after seeding. You can put it on the soil surface anywhere. It's not volatile. And we have broad-scale boron deficiencies. It looks like across the prairies, and it seems like almost globally. So it's something we're looking at very closely all the time. Toxicity from urea. The tips are sensitive because they're not really covered very highly and not protected because they're used to sense things. This is urea that I used a lot here to kill off the seminal roots. You can see that the shoot still is making it because it's living off of the seed. And the remaining seminal root just put out a lot more lateral roots trying to make an effort to feed this plant. But this plant will never be a really productive plant. And you won't even know why there's so much mortality during the growing season as plant competition starts to grow. And the plant can do a lot of things in the soil. It can change length and angle. It can put out change the numbers. It can change the density and length of the root hairs, and it does all those things again genetically, expression of regulation of transporters as well. So where are the roots? Everybody asks, where are the roots? Well, here are a lot of crops, and this is done in southern Saskatchewan, the Swift Current at the research station there, looking at, and this means that 50% of the roots are in this depth. So here you go, 15 centimeters, 6 inches. Pretty well, other than alfalfa, which we know goes down there, and chickpeas. The top 6 inches, I remember 50% of roots are. The remaining to get to 95% go way, way down. And then the max is a lot deeper, but you don't want to go here unless you have to. You don't want to go here unless you have to because it costs you, the plant, a lot of energy to grow that root down to get that water. And it looks as though it's the amount of moisture in the top 6 to 10 inches that determines how far the plant is going to go the root down. And that costs. That's why drought is so bad, because the plant has to go down for water or everything stops. So let's take a look at roots. Here are the kilometers of root per meter squared. Barley, not bad. Faba beans, poor. Maize, pretty good. Potatoes, not a huge root system in there. Look at rape or canola, 28. Look at soybeans, not much. Look at wheat. Why do we grow wheat? Why do we grow canola? 22 million acres of each on the prairies. And they grow them worldwide because they got good roots and they got good genes. Lots of diversity in genes and good roots. Here's how things move through the plant in reality. Here's soybean, young soybean and earlier soybean. You can see here the vascular system that things are moving in. And then at the node, we get a diversion into the laterals. Here's wheat, showing the vascular system. This is a cross section against the base, through the base of a soybean leaf. You can see the xylem here and the phloem running all the way from the root tip to here. Here's wheat. You can see the main vein and then the venules here. Get into the veins, to the tip, and it's done. One thing I'll point out is the tip of the leaf is the oldest part of the leaf. So when you see senescing taking place, you will see it starting at the tips because it has to use the veins to take everything back to the stem to remobilize it and move it if it's due to senescence. And so when you're seeing tips starting to brown, die off, depending on whether it's potassium or something else, it'll be either interveinal at the tips, and that's kind of why. Now here's where sort of diseases can have an impact. These are the stomata in wheat on the underside of the leaf, and this is just fungal damage here. And you can see that the stripe rust here is affecting the cells here. So it's interfering with photosynthesis. And as a result, we can generally get a fungicidal response, even if the disease isn't monstrous, uh, simply because it provides some health, plant health, around these small disease organisms and breakdowns that are taking place. The one thing we got to be careful of, it's with plant pathogens, is we didn't learn our lesson with insects and we didn't learn our lesson about herbicide tolerance, and we may not be learning our lesson about resistance to fungicides. But I sure wouldn't recommend going ahead and putting on a fungicide to get a small bump in production. It probably improves plant health, but the economics of it may or may not fit. We're looking at the concept of source and sink. So the source is the leaves, or it could be the seed in the beginning, but that's where the nutrients and the photosynthetes are coming from, and the sink is where it's going. So where's it going to? The seed, into the tissues and the roots. And if you think about the plant through its life cycle and what's going on in the source and sink, what are the shoots doing? What are the roots doing? If the shoots are doing poorly, it means that nutrients have to be pumped down to the root and you're going to lose productivity. If the roots and the shoots are doing well, you got the best chance of getting production to its maximum potential. It's a way to look at things sort of a little differently. And then again, once you have those photosynthetes, how does the plant move it? Well, here we go. Here's the source, here's the sink. The source is producing sugar. The sugar is passed through those plasmodesmata into the companion cells, and then through the plasmodesmata into the phloem. Now, as the solutes build, here's the principle. If you've got high solutes, water will move from a low concentration, from a high concentration to a low concentration. So as we build the solutes here, water moving up the xylem will move through these portholes and pressurize this, putting pressure on it, which then is relieved here as the sink is taking those nutrients. So the solutes decrease, they're utilized by wherever the sink is, the water goes back into the xylem, and up she goes. So we recycle the water. You can see it right here. We can move it around, we can move it to the roots, we can move it to the seeds. The plant can move it in the phloem both ways, because sometimes the photosynthetes are moving up towards the shoot, and sometimes you're moving down to the root, depending on where the plant is in its cycle. And that water transpiration moves up here, and it pulls those nutrients up to the top of the plant. And that's how it moves them around. You take a look at this. When does it do that? Well, the plant goes to sleep here at night, shuts its stomata, uses some of the sugar that it produced for respiration during the night because it has to stay alive. But come morning, when the sun comes up, the blue light stimulates the stomata and the chloroplast in the stomata to produce a little bit of energy, which then opens up the stomata. So the potassium level rises. And we can see that the osmotic pressure at 7 in the morning is low. The osmotic pressure increases, so that keeps the stomata open here. Uh, Eventually, the potassium starts dwindling, but the sugar production continues because now the stomata are open and things are rolling along, and then towards evening it shuts down. So a tip, if you want to put on anything that you want to go in through the stomata, you want to do it when they're wide open. Otherwise, you do it late at night and they're closed, you're not going to get it in. You may get diffusion into the plant, or you might get access into the leaves, but you probably won't get much in through the stomata. And after it's dried on, I don't think you're going to get a whole lot on through the stomata, unless it's right on the stomata. Nutrients. Not going to spend much time on nutrients because I'll cover them under the individual crops. Take a look at this. 96% of everything in the plant comes from carbon, hydrogen, and oxygen. So, what's the most important nutrient for a plant? Carbon, CO2, because that's the source of the carbon. So, you know, we're worried about climate change for sure, because it's always changing. But carbon is really key, so we want to get that CO2 in there. We want to keep those stomata open as much as we can, so stressful situations where we get that closed leads to poor crops. Nitrogen, I'll list these. Nitrogen is the highest amount in here, followed by potassium, then by calcium, then by phosphorus, then by magnesium. A lot of people don't realize that potassium is so important in the plant. It drives everything. It's not used much in molecules and proteins and so on at all, but it's used to pressurize things, to regulate things. Anything that moves, like a stomata, like a root, like a leaf that expanding, is all done by potassium. And we're starting to see potassium deficiencies, especially later in the growing season when it gets dry. And basically, when it gets dry, it's hard for a plant to get anything. That's why regular water is really, really important. We won't talk about this. And the transitional metals, we don't use a lot of them, but they're really key to all kinds of processes. And you guys can look at this. You can't do much about it, but mass flow are those nutrients that move a lot in water, like calcium, like sulfur, or sulfate, like nitrate moves with water. Phosphate doesn't move in the soil. Potassium doesn't move much. Manganese doesn't move much. Molybdenum is an anion, negatively charged. The soil is positively charged, so anything that's negatively charged will move a lot easier. Boron, for example, is another anion. Sulfate is an anion. Nitrate's an anion. So that's how you can sort of utilize that if you want to know how it moves. We already talked about pH, and staying in this range is a pretty good place to be. If you got land that's between 6.5 and about 7.5 or 7.3, you would just about died and gone to heaven in terms of soil quality, just from a pH perspective. Uh, we talked about these that are moving in the soil, but in the plant, it's a little bit different. Uh, in the plant, these nutrients, along with probably molybdenum, are mobile. N, P, K, and S. Magnesium are all remobilizable as the plant is deficient and as the plant is senescing. It will extract those. So nitrogen, phosphorus, potassium, magnesium, molybdenum, and chloride are negatively charged. Magnesium isn't, neither is potassium, but the plant will still mobilize it. Immobile ones are basically these during the growing period when, and you notice sulfur, a lot of people think that sulfur is mobile in the plant. It basically isn't. Once it's into products, proteins, and so on, it's not mobilizable until it's going into plant cell death, not easily mobilized. These nutrients aren't really remobilizable. So when you see tip problems and deficiency, those are the ones that are sort of not be can't the plant can't remobilize. You're going to see nitrogen, phosphorus, potassium, magnesium deficiency in the lower leaves. You'll see these in the upper leaves when there's a deficiency in the soil. And I guess I should mention that we don't have to worry about deficiencies if there's plenty enough in the soil. They will get most of those nutrients from the soil. The trouble is it's so variable across the field that certain plants get lots of it, other plants may not, and they're in the same field. So by fertilizing, by putting on foliars, you can make sure that basically every plant gets access to some good nutrient quality. This one is very common. Most farmers will know this, but this just demonstrates there are interactions between nutrients. So you don't want to be going and pouring on, a lot of phosphorus or a lot of nitrogen, especially when you're not balancing it off with sulfur. So you can study that and talk to your agronomist about that. One of the last things we'll talk about here is how plants remobilize the nutrients, or when they're senescing, how do they do that? Most of it comes out of the chloroplast and the starch and so on in the chloroplast. And when we get starvation of the plant, we get senescing going on, and we get photo damage, we start breaking down and we get autophagosomes that form, and we will break off chunks and we'll move them into the chloroplast through something called chlorophagy, where it's actually breaking down the chlorophyll and moving it into the vacuole, where it will be degraded and make it mobile, especially going into plant cell death. When the plant is senescing, it will move almost everything to some degree. When it's just deficient early in the growing season, it won't go and move everything, and that's why we get differences in mobility of nutrients depending on the growth stage. And pretty well all plants have similar growth patterns. I said they're pulling up a lot of nutrients until we get to the flowering stage, and then it kind of tapers off. And that's a general trend that you have. Here's, we know certain plants need a lot more of something or other, and this is sulfur and canola. And we know that through the growing period, the demand changes. For example, in the vegetative stage, these are the kinds of requirements in general for plants. When it goes in the reproductive components, we get a big uptick in all involved in pollen tubes, and all pollen tubes, lignin and pollens. I'm not sure what the PS stands for. Lignin and pollen, stigma and pollen tubes. Those all are utilizing much more. That reproductive stage pulls a lot of nutrients from the plant, and it's very critical in terms of loss of embryos going into that reproductive stage if you're short of carbohydrates, the sugars that are required for growth. And nowadays we're breaking down things like this by biochemical functions: nitrogen, sulfur making proteins, phosphorus and silicon for energy, these for osmoregulation, enzymes and cofactors, and these transitional metals and micronutrients for electron transport, ATP, photosynthesis, respiration. I won't go through this because we'll be covering these in each one of those crops. So I hope that gives you just a touch. We're into the last stretch in terms of stress. So let's talk about stress. And I've mentioned this before. When we have no stress, we get abundant resources, cell division and expansion, and that's what leads to a crop. When we get stress, we get events and damage repair instead of expansion. The plant is expending so much energy on damage repair, can't move nutrients, can't open its stomata, so it's being cooked in the sun, and as a result, we get resource limitation and cellular damage, and we get growth control. That's about as simple as it gets. We're either in productive mode or we're in defensive mode. And we know defensive modes from abiotic stress are killing us. Most of the losses in terms of crop potential productivity is related to abiotic stress. Even we take, corn, we figure the average yield is about 24% of the maximum at most. Wheat is about 13%. There's lots of room. You can see the genetics in wheat. We've got lots of room, and yet we're making slow progress in wheat genetics in terms of improving things. So we know that abiotic and biotic stress are important, but mostly abiotic stress. So how do plants deal with that? Abiotic, cold, heat, chemicals, the same way, basically. Receive a signal, gene activity, stress tolerance. Already seen this. The one thing I'm going to focus on, because it's probably the most important one, and every research paper you pick up today is going to be talking about ROS, reactive oxygen species. And they're generated in the cell because electrons leak during photosynthesis and respiration. So as important as photosynthesis is, and respiration in the mitochondria, they also have problems when there's too much sun and not enough water, basically. Excess reactive oxygen species are produced and they attack the cell. And the same thing happens in human beings. Sometimes they're called free radicals, but they damage the cells in the same way. And it's a double-edged sword, because if you get excess reactive oxygen species, it causes cell injury, cell death, and oxidative stress. High levels cause stress, molecular cell damage. We get scavenging by antioxidants that starts to occur, and we get impaired physiological functions. Sort of cover that in the general way. When we have low levels, though, the plant uses these reactive oxygen species as a signaling molecule. Cellular growth and development, they're signaling growth. They're signaling adaptation to the environment. Is there stress or isn't there stress? And they're scavenging the system so they can keep it in balance. When a disease is responding, or when the plant is responding to a disease attack, what it will do is it will upregulate or express genes that will produce hormones, for example, salicylic acid, jasmanic acids, other kinds of things that the plant can biosynthesize to defend it. And it may kill the cells around the attack using reactive oxygen species to prevent the spread of it. It will also line up a battlefield of mitochondria, which can multiply quickly to try and defend it, or where there's a shortage. So the plant knows what to do, but reactive oxygen species are a problem under severe stress. Here we go. Lipid peroxidization. It attacks the cell, and that plasma membrane with the lipids in it, it destroys them. It destroys nucleic acid. It modifies amino acids and proteins and enzyme activity, and it buggers up, for lack of a better term, polysaccharides and sugars in the cell wall, leading to cell death, growth reduction, and yield loss. The variability in the field of all those factors we talked about causes the production of reactive oxygen species which damage the cells. This looks complicated, and it kind of is, but it tells a story. So where does that take place? We said in the electron transport, right? In the chloroplast. And it produces, these sods are one of the enzymes that are really active, and I'll cover that quickly, and so are these. Produces hydrogen peroxide. Sod here in the mitochondria, what we talked about. And we get this cycle that is used, which is ascorbic glutathione cycle. Ascorbate is vitamin C, and I'll cover that. And we get peroxisome damage. That's where the hydrogen peroxide is destroyed and effectively nullified. So this is all going on in the cell with all these kinds of things inside the plasma membrane here. And the plant is dealing with that. So here are the non-enzymatic approaches: carotenoids, those colorful things, proline and amino acid, alpha tocopherol, vitamin E in the oils of canola, ascorbate, vitamin C. All plants produce vitamin C to deal with antioxidants, and we take vitamin C for the same reason. We take glutathione for the same reason. It's a sulfur compound. And we have all kinds of alkaloids and phenols that are involved in non-enzymatic ways to scavenge these free radicals. Then we have the enzymes. Remember I said SOD right there, catalase, ascorbate peroxide, glutathione. Now all the enzymes that are involved in that. And we get the free radicals destroyed. And this is the magic. Manganese SOD in the mitochondria, iron SOD in the chloroplast, and zinc copper SOD in the chloroplast and the cytosol, and there's a nickel one as well. Those are the ones that help the plant get rid of those doggone reactive oxygen species that occur when the sun is shining bright, there's no water, and the plant is cooking. The other ones that are important, potassium. Remember I said potassium moves things. It pressurizes things. It opens stomata. If you're short of potassium, the plant can't respond, improves ROS balance and antioxidation. Take a look at that. Do not let your plants run out of potassium at any point, nor sulfur at any point, because those are the scavengers that work the most in humans. Glutathione is key responder to reactive oxygen species, which occur in our cells in the electron transport system. So remember, we're all eukaryotes. We behave the same, just, think like a plant. And these are the major nutrients that are involved in that scavenging. We talked about all of those, the iron, magnesium, boron, calcium, sulfur, and K. Do your best at getting those into the plant. But if you don't measure it, you can't do anything about it. Start doing tissue testing if you're not. For sure, do soil testing if you're not. And start doing genetic testing of your soils slowly over time so you can start learning about what those microbes are doing in the soil, and we can understand the diseases and the good guys. Last 2 slides. And I bring this out, Dr. Kaplan, who was retired at the University of Calgary, did a lot of work in terms of using nutrients and especially micronutrients to deal with childhood mental problems. And I want you to take a look at this. What are the elements that are involved in this pathway to produce serotonin, one of the mental sort of hormones? Iron, vitamin B6, copper, vitamin B6, molybdenum, iron. We just talked about some of those things. Here's dopamine, vitamin B6, zinc, vitamin C, magnesium, vitamin C, copper, vitamin B5, manganese, folate, zinc, B1, 6, B3, B6, vitamin C, iron. Right here, iron, B2, 3, and so on. Those transitional metals are involved in the processes and the enzymes in all the eukaryotes. All of them need it every day. And I leave you with this. Think of the long-term implications of feeding the livestock and the urban feedlot. What do those mean in the long run as we extract those nutrients from the soil and concentrate them? Well, thanks for listening. I hope you're still here and still awake and understood most of this. Don't worry too much about it. Like I said, you can't see it. But at least you know that this is going on and have faith that the plant knows what to do. Hope you enjoyed it. Thanks a lot for listening.